Hello there, Dan Guerra from VerEvMed, bringing you another exciting lecture over YouTube. Today we're going to be finishing, I think, that arc on reactive oxygen species by bringing it and wrapping it back up towards the aging discussion that we've been having for the last several episodes of these VerEvMed lectures. So I want to get started right away so we can get this done and you can get on with doing what else you were going to do today after you get this stimulating lecture. So let's get started. Again, we're going to talk today about reactive oxygen and aging. This is lecture series number 4 if you're keeping track. This is part 3. Oxygenases, peroxidases, and function. Notice I use categories of 4 and 3 like my favorite philosopher, Immanuel Kant. All right, hydroperoxidases include 2 sets of enzymes, catalases and peroxidases, or frank peroxidases. Peroxidases reduce hydrogen peroxide at the expense of several other substances. So you take hydrogen peroxide, a reduced substrate, you make 2 molecules of water, and then you make the oxidized substrate. This is a very common procedure in fatty acid metabolism. Now, catalase, the second form of hydroperoxidase, actually uses H2O2, hydrogen peroxide, as an electron acceptor and electron donor, depending on which way the reaction moves. Okay? And that's just, you see the reaction there. Hydrogen peroxide goes to water. Now, where do you find some of these reactions? Well, notice the word peroxidase, okay? So that's peroxidase. That means that you have one substrate that's going to be involved in the reduction or oxidation, and the other substrate is going to be involved in interacting with hydrogen peroxide. And usually water will be one of the products. Those reactions, the one that comes to mind, and I guess I don't know why, are those reactions that are involved in fatty acid metabolism in the peroxisome. Peroxisome is a very important organelle that gets terribly overlooked in most cell biology courses. But the peroxisome is a favorite of us lipid biochemists because that's where some really important reactions occur. Removal of fatty acids from the plasma membrane and breaking them down to shorter chain fatty acids so that they can ultimately beta oxidize for energy or build back up via elongation reactions. All that's going on in the peroxisome. So that's really important because the refurbishing of the lipid content of the plasma membrane is vital for plasma membrane integrity. That is its structure-function relationship. That is all of the signaling that goes on across the plasma membrane requires fatty acid removal, specific fatty acid removal, and then resynthesis of those fatty acids in specific stoichiometric geometric configuration. It's a highly complicated procedure that it is necessary for the peroxisome to be involved. So peroxisomes get their name because when they were first discovered, people noticed that there was, a lot of hydrogen peroxide in them. So this is kind of a really brief summary of peroxisomes. Interestingly, unlike all organelles except the mitochondria, peroxisomes actually reproduce autonomously in the cell based on need. They also go through a process called pexophagy, just like mitophagy, where the peroxisomes actually disintegrate or break themselves down via autophagy, just like the mitochondria do, when it's no longer necessary for those organelles to exist. And sometimes when those cells that harbor those organelles, mitochondria and peroxisomes I'm talking about, are generating too much reactive oxygen, then the organelles that are responsible for all that reactive oxygen, and that's where you make a lot of it in the peroxisome and in the mitochondria, that they eliminate themselves via autophagy. That is, they consume themselves. Okay, so it's a really important process of the cell. It's a dynamic process of all particularly mammalian cells, although plants also have peroxisomes, as do invertebrates and lots of other life. Okay, so let's get started with this. They're self-assembling then, that is that they're autonomously replicating. They don't last very long. They probably arose during evolution because of the increase in oxygen in the atmosphere, because that's what they do. They really are responsive to molecular oxygen. Interestingly, they have no genome. Peroxisomes don't. All the proteins, therefore, are going to be coming from the nucleus. They're all going to be translated into cytosol. And there's going to be something called a peroxisome targeting sequence, or a PTS, that directs them to that peroxisome. There are multiple types of these PTS, but if you're going to make it into the matrix of the peroxisome, you're going to have that motif, serine, lysine, leucine, at the carboxy terminus. PEX5 is a particular protein that has a PTS1 receptor, and that directs cytoplasmic proteins to peroxisomes. So you have to build the outer ornamentation of the peroxisome with peroxisomal receptors so that they bind to the peroxisomal target sequencing for enzymes, for example, that we're going to visit here, that exist in the matrix of the peroxisome. So it's a nice, complicated, but well-organized set of conditions. There's a lot of other peroxisome targeting sequences, so not just that one. Membrane PEX proteins have a different targeting sequence altogether. So if you're going to just make it into the membrane of the peroxisome, you're going to be targeted totally differently than if you're going to make it into the interior of it. So there's a lot known about this. Here's just one more brief discussion of it. You start off with a precursor membrane that's synthesized probably from the endoplasmic reticulum, maybe also the mitochondria. Peroxisomal membrane proteins start building in. These are, again, translated from soluble ribosomes in the cytoplasm. PEX19 interacts with PEX3 and PEX16, starts forming these outer membrane proteins. We call this a peroxisomal ghost. Then peroxisomal targeting sequences bearing matrix protein like PEX5 and PEX7, which has the second targeting sequence, interact with PEX14 and all these other PEX proteins, and now you start moving proteins into the peroxisome. You're maturing it. You see, now you have catalase. That's the showing you an example of the kind of enzyme you're going to get in a mature peroxisome. And then with PEX11, the help of PEX11, you go through fission reactions, and that's how you divide. Now you make more peroxisomes. You make more peroxisomes when you need more. When do you need more? When there's a lot of turnover, for example, of polyunsaturated fatty acids in the plasma membrane. This happens during vital times, during excessive amount of biological activity at the membrane. Okay? Such as, for example, during an immune response. Okay? Oxygenases, of course, are another type of enzyme that is going to be involved. These are all redox enzymes, right? They catalyze, we already mentioned this in a previous talk, but this is just a recap in case you didn't see it. Of course, I'm sure you did. Catalyzing incorporation of molecular oxygen into substrates in 2 steps. Oxygen is bound to the active site of the enzyme. Okay, so usually there's going to be involved some kind of flavin nucleotide, or maybe an iron, or even sometimes a copper center, usually an iron, heme. Binds oxygen and it reduces it, and then or that process is associated with transferring the oxygen to the substrate. 2 sets of enzymes. You need the dioxygenase that incorporates both atoms of oxygen, shown here. You've got a substrate, there's molecular oxygen. Now you've completely oxygenated that compound. That's, for example, in those enzymes that make oxygenated fatty acids, such as the eicosanoid family. Eicosa means 20. And those include things like prostaglandins, right? Oxygenated long-chain polyunsaturated fatty acids like the eicosanoids get oxygenated. Those oxygenated compounds are called eicosanoids. One class of them are the prostaglandins. Autocrine hormones, very important in the cell. Another type are called monooxygenases. They incorporate only one atom of oxygen to the substrate, and the other one ends up going into water. That is, it gets reduced with hydrogen ultimately to make water. And so that means you're going to have a reductant here, and that reductant can be either NADPH or NADH. Later we'll see where NADPH is really important. Oh, sorry. There's just a reaction. You've got a reduced substrate, molecular oxygen. You've got some other substrate here. We're just putting a Z in front of it. Now you're going to make a hydroxy compound. Okay, these are going to be really important in fatty acid metabolism. You're going to make water, and you're going to make the oxidized co-substrate. That's probably going to be something like, guess what? NADP. This could be NADPH. This could be a fatty acid, this molecular oxygen. You get hydroxylated fatty acid, excuse me. You get water formed, and then you get NADP, okay, for example. All right, let's take a look at fatty acid metabolism briefly here, because it's sort of a central player in what I want to get into later when we talk about these processes. But it's also a central player right here and now. And of course, it's also because fatty acids are just so darn interesting. Even after they're synthesized, there's just so much metabolic play, even dietary fatty acids, which make up, of course, the essential fatty acids, which, of course, are linoleic and alpha-linolenic acid. Right? Okay. So you got esterified fatty acids bound to some membrane lipid. Okay. The enzymes that can remove those fatty acids are called phospholipases. There's a whole family of them. One in particular that's involved in stress responses, particularly those responses involved in inflammation, is phospholipase A2, shown here. So phospholipase A2 itself is regulated, and there are inhibitors of this reaction. But ultimately, for purposes of just getting through this, all we're going to say is we make free arachidonic acid. Arachidonic acid can be reacted upon immediately, right in the membrane, with lipoxygenases. We call them LOX, L-O-X, all caps, or cyclooxygenases. We call those COX, C-O-X, all caps. Multiple forms of COX, multiple forms of LOX, depending on substrate specificity, and then, of course, product distribution. One key product of LOX are leukotrienes, really important vasoconstricting compounds in the cell. They're actually ones strongly associated with the pathophysiology of asthma. And then if you go through the COX reaction, of course, a common product, not the only one, just like this isn't the only one of this enzyme, are the prostaglandins. Again, these are autocoid hormones. Prostaglandins, of course, really important in human reproduction, amongst other things. These enzymes are the targets of a lot of pharmaceutical drugs and over-the-counter drugs. Cyclooxygenase, in fact, our friend indomethacin will inhibit that reaction competitively, and also aspirin will acetylate that enzyme on a serine residue and will act as a suicide inhibitor and kill that protein. So that's where aspirin functions at the COX level. Now, that's not enough. Arachidonic acid can also be reacted upon. These are all, again, enzymes that are redox associated. Okay, LOX, COX, now epoxygenases, and also omega hydroxylases. Okay, the omega hydroxylases, take a look at these first. They're going to make a series of compounds that start off with 20-hede. 20-hede is a really important compound. That's going to be an hydroxyicosa tetraenoic acid. Hydroxyicosa tetraenoic. Isn't that wonderful, right? Tetraenoic, what does that mean? 4. Ene is, of course, double bond. That's going to be a product that's got 4 double bonds. It's going to be hydroxylated. It's going to be 20 carbons long. Okay. Now, there are multiple forms of this one compound, and they're inhibited, feedback inhibited by products of the reactions. But ultimately, what hedes do is they're involved in vasoconstriction. Really important. Vasoconstriction of what? Things like smooth muscle cell. Okay. All right, epoxygenases, totally different pathway, totally different type of reaction. You're making epoxy residues here. It's a different process than hydroxylation. Again, they can be activated and inhibited by all kinds of series of drugs and naturally occurring compounds. Okay, there's phenobarbital there, positive activator of epoxygenase, for example. Interesting, right? Now what we're making are epoxyicosa trienoic acids, okay, and sometimes tetraenoic. So epoxyicosa trienoic and tetraenoic fatty acids, right? So you got the epoxy residue, COC bound, right? Oxygen above the plane of the hydrocarbon chain, of course. Okay, so these epoxides now can be further metabolized to dihedes, right? Again, we're getting back to being hydroxylated forms, but here we got 2 of them because we went through an epoxide hydrolase. So we removed water, we got rid of that epoxide. We made what? Well, yeah, we made hydroxy because of that. And what do we get here? EETs rather than HEDs, EATs, are involved in vasodilation. So depending on which way you go, epoxygenase or the omega hydroxylase, you're either going to cause vasoconstriction or vasodilation at the local level. These are local hormones, but they are indeed hormones. Okay. All right. So that's just a little idea. And I know this is getting more and more into this, and I know you like it. Here's a paper that was published, oh gosh, already 5 years ago. It's so sad how time flies. But Journal of Biological Chemistry papers, they stand the test of time. They usually are not refuted, and if anything, all they are is added to. Again, it's an oversimplification and generalization, but in general, JBC papers are just excellent. If you read a paper in JBC, you can be pretty sure about that evidence and pretty sure that the verification of the evidence is going to be right in that paper. All right. Now, calcium and magnesium and all kinds of other responses, such as simple stress or trauma, can induce phospholipase A2. Now, this is a particular form, gamma, right? And I means inducible, okay? Like a little I always means inducible. Now, depending on how this enzyme gets mobilized, it's either going to be a PLA2 or a PLA1. You're going to make a lysophosphatidic acid, this pathway. Lyso just means you've removed a fatty acid. Lyso means removal or absent. And you're going to make a arachidonic acid. That's a 20-carbon fatty acid with 4 double bonds of the omega-6 series. Common fatty acid in the 2 position of preformed membrane phospholipids. Arachidonic acid, then, doing this cascade, is going to go through those enzymes we just looked at. LOXes to make HEDs, COXes to make prostaglandins, and whoa, here's another one, the cytochrome P450s, and that are actually, those are the enzymes that we were calling just generically epoxygenases. Not all of them are epoxygenases, okay? But the cytochrome P450s are a tremendously big enzyme family. And they're involved in drug detox, drug metabolism. Sometimes they cause drugs to become more toxic. They're involved in steroid metabolism. That includes things like from steroid pathways like vitamin D metabolism, right? I could go on and on and on. And in fact, later in the talk, you'll see that I do go a bit on and on about these. These are really interesting enzymes, the P450s. But they are basically detox enzymes. You find a boatload of those in the endoplasmic reticulum. You also find some, of course, in our friend, the peroxisome. All right. You can also make these various forms of lysophosphatidic acids because PLA1 is activated here. That's going to remove the fatty acid from the 2 position, but the 1, that tends to be a saturated fatty acid, which can go into fatty acyl-CoA pools. Fatty acyl-CoA pools in the mitochondria will inhibit this enzyme. Okay, so it's just an aside. That's an acyl-CoA synthase. Don't worry about it for now. You can further metabolize these arachidonyl lysophosphatidylcholine, that's what that PC is. That's PC there too. It's just like a stand-in for all phospholipids, but PC is common in the plasma membrane, of course. And you start forming actually endocannabinoids. There's 2 arachidonyl glycerol, right? And this is a pathway that ultimately can lead to things like anandamide, which are endogenous cannabinoids. Here's a lysophospholipase D, which is going to give you 2-arachidonyl lysophosphatidic acid. What we remove there, of course, what? We remove choline, right? So all we're left with here is just a phosphate ester there at the 3 position of that glycerol moiety. Okay, so lots of interesting things are happening simultaneously when we're also looking at these reactions. So I want you to get the feel for how absolutely bloody complicated lipid metabolism is, because a lot of reactive oxygen is synthesized and quenched and utilized in fatty acid metabolism, as well as in prenatal lipid metabolism. So it's a big thing in the cell, right? Fatty acids. Now, here's the reaction itself, taking the arachidonate, our friend, the omega-6. Remember omega-6, that's where the last double bond is here. So this is the carboxy terminus. If that double bond is 6 carbons away from the carboxy terminus, we call it omega-6. If it's 3 carbons away, we would call that an omega-3. So the omega means N, the end of the fatty acid is a carboxyl group, carboxylic acid. So that's where you're going to be counting it from. Okay. All right. Actually, this is I got that reversed. You should be talking about the methyl groups. This is the omega-6 from the methyl terminus, not from the carboxy terminus. But this looks like that, but actually it's the methyl terminus where we count omega-3 and omega-6. Sorry about that. All right. I hope I didn't confuse you, but we'll clear it up later. Don't worry. Anyway, phospholipid then, phospholipase A2 makes a arachidonic acid. Arachidonic acid used in cyclooxygenase reaction makes prostaglandins. I told you that aspirin inhibits that. Also, indomethacin and phenylbutazone. Lots of over-the-counter drugs will inhibit this reaction. And the reason you inhibit that is like an anti-inflammatory response. Because when you make prostaglandins and all these other oxygenated fatty acids, this is on the way to inflammation. Okay. Now, the actual phospholipase A2 is inhibited by things like corticosteroids. Okay, so that's anti-inflammatory too. Now, here's the P450 monooxygenases. Boy, look at all of these enzymes. They're called CYP for short, but look at all of these enzymes involved in drug metabolism, arachidonic acid. There's thromboxane, which of course are oxygenated from the COX pathway fatty acids. There's one particular CYP there. Bile acid, okay, necessary to have cytochrome P450s. Lots of steroidogenesis in the brain involves specific CYPs. There's prostacyclin, steroid, vitamin D, retinoic acid, and on and on and on. Okay, all cholesterol metabolism itself, synthesis of it. You can't go wrong if you say that CYPs are involved in lipid metabolism, and lots of drugs are actually lipids, and that's probably why there's so many that can join here. So human cytochrome P450s, we're not going to talk hardly anything about this today, but my colleague will in the future talk about drug metabolism with specific P450s. But here's just a nice standard run of lipids: cholesterol, steroids, bile acids, vitamin D, fatty acids, retinoic acid, vitamin A. All of those utilize cytochrome P450s. By the way, they're called cytochrome P450s. I know that you've been wondering why. Because they have a lambda max. When you measure the protein in an aqueous solution, you're going to get an increase in absorbance on a spectrophotometer at 450 nanometers. So we call those P450, proteins that absorb at 450 nanometer light in a spectrophotometer. Now that's because of that flavin residue, why they have that lambda max there. But that's why they're named that. So now you know. Here are various classes of them. Some of them have flavonoidine dinucleotide, iron sulfur centers. Notice NADPH is involved in class 1 or NADH. Right? Class 2 has an FAD-FMN. Then there has to be electron transport to the heme residue. The heme is the actual carrying agent of the enzymatic reaction. The specificity of the enzyme is the heme part of the protein, where substrate goes to, here it's going to be going to a hydroxylated product. Class 3, you've got FAD and FMN like you do in class 2, but you have it directly linked up to the heme. Once again, there's your substrate to product, and there's class 4, that is an iron sulfur center. Okay. Now, here's some more about nomenclature. Cytochrome P450, you call them CYPs. That's simple enough. Family, the largest designation, right, in hierarchy. All of the proteins that are going to have the 3, about practically greater than 40% of all the CYPs have a 3 there. Subfamily is going to have a letter designation. That's going to be about 60% of all that subfamily. And then the last is going to be a number again. You like that? Number, letter, number. Pretty simple stuff, right? And that's the specific isoform. So you can see that these enzymes have a tremendously large family. So much that we have to have this tremendous, 4-level designation, right? Okay. There are a bunch of them. Look at this one here: drug metabolism, 2 subfamilies, drug and steroid, vitamin D. Let's take a look at just CYP4. That's going to be involved in arachidonic acid and fatty acid metabolism. Okay, it's just key in on that one. All right, this is from a paper published about a year ago. This is the end of April 2017, so about a year ago. Excuse me. This is in the AJP, American Journal of Physiology, particularly the Heart and Circulatory Physiology Journal. And what does it say? Okay, 20-hydroxyicosa tetraenoic acid. Remember I told you before, 20-hedy, right? Is the omega hydroxylation metabolite of arachidonic acid. We saw that already. It's made by CYP4A and CYP4F, and NADPH isn't required. 20-hedy is a pro-inflammatory oxygenated fatty acid. Okay, pro-inflammatory. Okay. And it regulates, look at this, vascular and renal function. I told you these were autocorrect hormones. So it's not that they're pro-inflammatory, but it's like these are necessary for the cell. So later on, we're going to see where people are trying to manipulate the levels of these, actually decrease or increase. Remember that these are necessary for normal physiology. So there's 20-hedy, plays a critical role in the pathogenesis of systemic hypertension. Wow. Renal stenosis, another bad pathology. And if that's not enough, atherosclerosis, okay? Lots of pathogens associated with one hydroxyicosa tetraenoic fatty acid, okay? 20-hedy is increased by hypoxia, removal of oxygen, and in fact, it inhibits hypoxia-induced pulmonary artery constriction, okay? Now, sort of paradoxically, okay, 20-hedy is involved in promoting prolonged hypoxia-induced pulmonary vasoconstriction. Prolonged, okay? So it promotes that, right? So these compounds will work one way or the other depending on what receptors they bind to and depending on the equipoise of the membrane in terms of electrochemical potential of the membrane, whether or not they're going to promote or inhibit a specific major global physiological response like constriction. Now, here's a key kicker here. I told you that these are NADPH dependent. Where did we get that from? Well, I know all of you have recently taken a biochemistry class, so I don't need to tell you for the first time, but just a reminder, glucose 6-phosphate dehydrogenase, of course, that's what? That's an enzyme in the oxidative pentose phosphate shunt, of course, the alternate glucose oxidation pathway in eukaryotic cells. What does that pathway do, by the way, real quickly? It makes NADPH. It makes carbon dioxide. That is, you turn a C6 into a C5. Whoa, what's that C5? Well, a major product of the oxidative pentose phosphate shunt is ribose 5-phosphate. So actually, this is where you make ribose for making nucleotides. Okay? Important pathway, but really important in terms of intermediary metabolism and particularly germane to our discussion today is production of NADPH. Okay, that is the major source of NADPH. There's actually two dehydrogenases in the OPP, and they both make NADPH. That's the major source of NADPH in the cell, is the OPP. Okay? So cytochrome P450 monooxygenase enzymes are functionally coupled in vascular smooth muscle tissue. So what's interesting is that this enzyme expression and the expression of particularly P450 monooxygenase, guess which one, the one's going to be involved in this 20-hedy production, are both co-expressed and found in the same tissue, vascular smooth muscle tissue to be precise. So let's take a look at this now. Here's your membrane. Now there's some recommendation from the literature, which can be verified, that 20-hedy is not simply an autocoid. It might actually get mobilized into cells. There might be a possibility of that because there are receptors for it. At any rate, we don't know that, so we'll put a question mark there. But we do know it's synthesized in situ, okay? And there's your CYP4 there, right? There's your enzyme that's going to make it. And now what's that going to do? It's going to increase the amount of reactive oxygen, okay? Now, with reactive oxygen, what you're going to do is you're going to turn on MAP kinase. MAP kinase is going to turn on ALK1. ALK1 phosphorylated, that's a phosphorylation there, is going to make it to the nucleus, and it's going to act as a transcription factor. Actually, it's going to function as a de-repressor of transcription for tumor necrosis alpha. So tumor necrosis factor alpha gets synthesized now because of this cascade system. Now, what is that? That's a major pro-inflammatory cytokine. This is a pro-inflammatory pathway. Now, there's glucose 6-phosphate dehydrogenase making NADPH utilized for that reaction, okay? Now let's take a look at what this business is going on down here real quickly by reading the narrative. 20-hedy caused an increase in mitochondrial reactive oxygen species, Showed you that. Okay. And it causes a decrease in this, what's this here? This interfering RNA here. Okay. This interfering RNA here causes a decrease in miR-143, but it also causes p42-44 MAP kinase, that's this guy here, phosphorylation. Okay. Which then leads to increased expression of ELK1, increased expression of ELK1, there's your friend there, then leads to an increase in the expression of TNF-alpha, we just showed you that, transcriptional level, which can then block, here we go with the new stuff, protein kinase G1 alpha activity. Okay, it's going to block that. Where are we at here? Okay, in the vascular smooth muscle. Okay, now inhibition. Now let's talk about this. If you inhibit this enzyme, which means you're going to decrease the amount of NADPH in the cell. Okay, so here we go. Inhibition of that enzyme, glucose phosphate dehydrogenase. First reaction on the oxidized phosphate shown. It's going to deplete NADPH available to the CYP4 group of enzymes. Thereby, it's going to decrease the synthesis of 20-HED. Boom, there. Inhibition of this enzyme also activates the PKG because now it's active because you don't have this TNF-alpha kicking it down. Okay, that happens without an increase. That's not nitric oxide. I know that really looks bad because it looks like it could be nitric oxide because nitric oxide is associated actually with guanylate cyclase type activity. It's not what this what the authors chose to do to make this particular cartoon. So sorry, it just says you don't, in cases where you de-inhibit this enzyme, you don't need to have cyclic GMP. That's what that means, without increasing it, you see. So no guanylate cyclase activity has been induced, which then inhibits 20-HED mediated increase in mitochondrial ROS. Why? Because now what happens is that you've got what? You've got no more glucose 6-phosphate dehydrogenase, you've got no more NADPH, no more CYP4 activity, cytochrome P450 oxygenase activity, no more HED production, no more active oxygen. Boom, you don't do any of this either. Okay? So blah, blah, blah. Now, increases, which then inhibits the 20-HED mediated increase in mitochondrial ROS. Decrease in mitochondrial reactive oxygen leads to an increased expression of miR-143. Now you get this kicked up again because that was suppressed, which then inhibits ELK1 expression. Boom, there's your block. Nope, no more of that. No more of ELK1. That's a kinase. Activation of, then that causes activation of a protein kinase G by the glucose 6-phosphate dehydrogenase inhibition also leads to a decreased ELK1 expression. That's also going to be involved in dropping that. Okay? Which in turn leads to a decreased expression of TNF-alpha. So the whole thing gets turned around, right? Turned from its laying on its back to laying on its belly. In other words, now what you're going to get, you're not going to get this cascade of inflammatory response. You're going to shut that down. One of the main reasons you're shutting it down, now, there's a lot of interesting things here about transcription and pro-inflammatory cytokines and whatnot. But what I look at is a bioenergetic, I put my bioenergetic cowboy hat on, and I say, well, how do you carry out these reactions? I want to know where you get reducing power. You need reducing power to carry out that P450 oxygenase. Look here, it's knocked down because you've knocked down the activity of glucose phosphate dehydrogenase. And that's really where it comes from. You don't have enough of this, you can't go anywhere, and you don't go through this pro-inflammatory pathway. Now, so let's take a look at this paper again. Okay, now here's the reduced abstract of the paper we introduced. Again, same paper, published about a year ago exactly. This is the last day of the month of April, I can tell you. Okay, 20-hydroxyicosate tetraenoic acid, our friend 20-HED, is produced. It can be produced. We saw there were other pathways that make it, but we're talking about the cytochrome P450 monooxygenase pathway, which is NADPH dependent. Now that oxygenated fatty acid is pro-inflammatory, and it contributes to the pathogenesis of systemic and pulmonary hypertension. So it's a bad player. Right? I like that term, bad player. Right? All right. Now, they tested in this research, okay, the hypothesis that inhibition of the dehydrogenase, okay, where the dehydrogenase prevents 20-HED production, right, and 20-HED production induces pro-inflammatory signaling that promotes secretory phenotype of vascular smooth muscle cells. So they want to test whether or not if you inhibit this, which we basically understand the logic there. No more NADPH, that you shouldn't get a 20-HED unless, of course, another pathway got engaged. But let's presume this is the main pathway here in these vascular smooth muscle cells, that you're going to get that decrease in the overall pro-inflammatory response. So the dehydrogenase inhibition and knockdown, they also did a knockdown experiment, decreased 20-HED levels. Okay, so that's good. That's sort of a proof of concept there. 20-HED then induces normally, remember these things, mitochondrial reactive oxygen, here they just say superoxide, activation of the MAP kinase. Again, then we're going down that pathway. What's that pathway? Pro-inflammatory cytokine pathway. It's going to also induce phosphorylation of ETS domain containing protein. That's the ELK1 that activates the transcription of TNF alpha. We saw that, right? And finally, 20-HED is involved in the expression of that, right? Because of the spectral transcription factors. Okay. So, inhibition of the oxidative pentose phosphate shunt enzyme, glucose 6-phosphate dehydrogenase, increased protein kinase G1 alpha, which reversed the whole induction process. Everything went the other way, okay? As I said, 20-HED repressed miR-143, right? That's interfering RNA 143, which suppresses ALK1 expression. So 20-HED elicited mitochondrial reactive oxygen and promoted secretory phenotype of vascular smooth muscle cells by activating MAP kinase ELK1, all of which can be blocked by inhibition of glucose 6-phosphate dehydrogenase. So obviously, that's a molecular target. All right. Now, here's kind of a pie chart looking at cytochrome P450 families. You see there's a great deal of them that are in the A4 and A5, 3A4 and 3A5 family, right? Not much of these here. Fair amount of 2Cs and 2Ds. Okay, so just give me an idea of the distribution. So, real quickly, CYP2D6 substrates. Look at all of these compounds they work on. Antiarrhythmics, antidepressants, antipsychotics. There's thorazine, beta blockers. I mean, these are some of the most major prescribed drugs. Analgesics like codeine and fentanyl, right? All of them are substrates for that CYP450 isoform. All of them. So these are really, really, really important enzymes in terms of prescribed drug, metabolism, right? Here are a bunch of compounds that are CYP3A4 substrates. Tylenol, there's codeine, cyclosporin, on and on and on and on. Valium, erythromycin, right? Now, again, when these enzymes, when the cytochrome P450s interact with these ingested drugs, sometimes, often, they then break them down so they're no longer biologically active. Typically, that's what happens. But also, sometimes they can produce toxic products. When they produce toxic products like acetaminophen, when it's broken down, it can actually cause what? Liver damage. Right? So remember, these enzymes, they're meant to detox the foreign compound because they look foreign and because they're metabolized that way in the cell. But often, not often, but frequently enough that it becomes important, drugs that are normally benign to the tissue or the cells or to the body can generate products after cytochrome P450 metabolism into toxic products. Okay? So that's an important thing to keep in mind. All right. Now, here we're looking at this production of a double bond, right? So we're looking at using a flavoprotein. This is going to be what? Right? This is going to be a hydroperoxidase because there's hydrogen peroxide, right? And you're going to be taking this saturated fatty acids. What that is, you're going to put a double bond into it. Okay. Now this is again going to be omega hydroxylation you're going to get to because it's at the end of the molecule, right? Well, it's at the carboxy terminal here. All right. So that's a really important reaction. Now, all of these enzymes, P4 are P450s. So there is PGH2. That's one of the initial products of the COX pathway. Cyclooxygenase pathway. When you're going to make prostacyclins, you need to have P450-8A. If you're going to make thromboxanes, you're going to make P455A. These are two just simple metabolites of originally arachidonic acid going through the COX pathway to form this PGH2, but look at totally different biological activity. Thromboxane is very vasoconstricting, involved in, for example, myocardial infarction, and prostacyclins tend to often be vasodilating, right? So once again, big difference depending on which pathway they go. And obviously that's going to relate to what cells are doing this metabolism, whether or not they're going to have high levels of thromboxane synthase or prostacyclin synthase. Now, I told you that these P450s work on bile acids. There's a common bile acid. Remember, that's a product of cholesterol. Here is cholesterol being metabolized through these very well-known testosterone, cortisol, estradiol down here at the bottom. That's what those are, okay? What are the enzymes involved in the metabolism of cholesterol to these sex hormones? Well, they are cytochrome P450s. And look at all the specificity there. So you can't make drugs that simply inhibit or modulate cytochrome P450s. You have to make drugs that are absolutely precisely specific to CYP isoforms. Of course, what's a good way to do that in terms of drug strategy? Well, one way that tends to work are either in specific inhibitors, so that you can find small molecule specific inhibitors. But because a lot of these reactions have very similar active sites, and because they have similar active sites, they have similar structure, and because they have similar structure, the inhibitors can kind of inhibit a lot at the same time with different levels of valence, of course. Then besides having outright inhibitors of these enzymes, monoclonal antibodies are sometimes used. Still run into sometimes a problem with cross-reactivity with monoclonal antibodies because, again, you're working on a structure, right, with monoclonal antibodies to bind to it and knock out the protein. So, but that's where drug discovery comes in. Just to give you, if that isn't enough about these cytochrome P450s, here we are making 25-hydroxyvitamin D. That's 125 dihydroxy, and there's just the dead end metabolite, 24-hydroxy. Vitamin D. So you see here how cytochrome P450s are involved in synthesizing the active forms of vitamin D and in the kidney by removing it, making the inactive. Okay? Depending on the kind of cytochrome P450 action. So very, very important to the cell. We're almost done here. All right, so let's take a look at what goes on in a general reactive oxygen motif. Hypoxia, altered insulin signaling. Where are we going to get this? Let me think. Diabetes. Calorie restriction, something we talked about in some previous episodes of VerEvMed discussion. Mitochondrial mutations, okay, that is mitochondrial gene mutations, which happen frequently. Physical activity, just simple physical activity, and also all kinds of compounds that could be in the diet or that are prescribed. All of them can lead to the production of reactive oxygen. And here are some of the ways that are involved in doing it. Okay, there's mTOR, targeted rapamycin. There's sirtuins, remember deacetylases. There's AMP kinase. There's the hypoxia-inducible factor. All those can be involved in the production of ROS. Now, when you take antioxidants, okay, such as ascorbic acid, vitamin C, or retinoic acid is sometimes used too, vitamin A, or in fact tocopherol or vitamin E, what are those effectively doing? Well, one way or another, either membrane or cytosolic, and working with glutathione peroxidase or working with NADPH, those antioxidants are going to try to remove reactive oxygen. That's why they're called antioxidants. And people take these all the time over the counter, right? This is in every pre-formed vitamin pill you can take, probably, unless it's a very precise vitamin, like this is just vitamin A, right? But in fact, vitamin A also acts as an antioxidant. Now, what ROS normally does is turn on all of this transcriptional control. NRF2, KEAP1, FOXO, all the FOXO genes, the heat shock factors, and the peroxisome enzyme systems, okay? All of that transcriptional regulation is controlled by an uptick in reactive oxygen. Okay. Now, what you get because of this buildup of ROS, because of all these events happening, you get then increases in antioxidant enzymes, trying to remove that. You're getting phase 1 and phase 2 detox enzymes, some of which are CYPs. You're getting heat shock proteins and uncoupling proteins. They're going to regulate gene expression and chaperoning of proteins in and out of subcellular compartments like the endoplasmic reticulum. Mitochondrial uncoupling proteins to be able to control the amount of ATP produced and changing biofuel from carbohydrate to fatty acid, for example. Lots of things like that can all happen because the reactive oxygen is induced gene expression. And when that happens, when all of that happens, you have a goodly amount of reactive oxygen, which are necessary to keep normal metabolism going. Normal metabolism, all of those functions I just showed you require some reactive oxygen. Which one did we just see a lot? Was all over the place, super abundant? Hydrogen peroxide. You got to make that, right? All right, but the cell repeals that issuance, right? The cell says, okay, we made enough of our reactive oxygen. We're going to now freshly transcribe genes. We're going to pull that back down. Now, the interplay between producing reactive oxygen and removing it is suggested to be paramount, maybe emblematic to a healthy, long life. That's an argument out there. Now, one of the reasons is because look what we're seeing here that are involved in this process. Caloric restriction and physical activity are pushing this. Don't we say that caloric restriction and physical activity are good things for health and for longevity? Well, it seems to be the case. So what we're talking about here is our complexities in cellular responses to stress. And stress is often good. So certainly caloric restriction stresses the body, right? Having long periods of fasting will stress the body. Likewise, physical activity will tire you out, right? How do you feel after you run 10 miles, right? You're going to feel pretty exhausted, right? How do you feel after you chop wood for 3 hours? You're probably going to feel pretty exhausted. Now, that again is built into the normal physiology of the human. Do a lot of exertion, create a lot of reactive oxygen, utilize that reactive oxygen to carry out a lot of endocrine, paracrine, autocrine hormone regulation of central metabolism, controlling inflammatory responses, mobilizing defense networks. All of that is necessary, right? All the time. You need a good defense to be able to withstand the vagaries of being alive in an oxygenic environment. But at the same time, it's a built-in repeal of that, removal of that reactive oxygen, so there's a balance. That's what I'm trying to show you here. This is mortality events, whatever they happen to be. They're going to be increasing, right? But you're going to have a period of time, if it's a linear process, where you're never going to be able to reap any benefits with reactive oxygen. And what this model suggests, this is primarily from animal work, and we can go deeply into this too, because there's a lot of interesting papers here, is that you actually get health promoting effects from exposure to reactive oxygen. That's what all this is about here. Health promoting effects of, for example, the cytochrome P450 oxygenases, the oxidase reactions, the peroxidase reactions, all vital to the cell, right? All those prostaglandins, all those leukotrienes, all those thromboxanes, all those prostacyclins, right? All those EETs and HETEs are all important for the cell. So the argument is, again, from modeling, right? Big hand waving here, but the argument is that maybe that's health promoting for the amount of time that you're exposed to ROS, but after a while, it does become a mortal event, that is a morbidity event, if you have longer periods of ROS reactive acid. It becomes harmful, you see? And so the argument is, is that the mitochondria is where they're showing all this ROS, that you have a factual mitohormetic response. That's what it's called in nutrition, right? Where the production of reactive oxygen is not really a linear process, right? It's got kind of this weird dip here where you get health promoting effects. And because you're utilizing this buffering system of reactive oxygen, you might always be lower. You might be gaining longevity, right? And a healthier longevity because you're always below the curve if it's just linear. And how are you going to be linear? Possibly by doping with high levels of antioxidants. All right, almost up. Here's a paper from Aging Research Reviews in 2010. Clinical applications in pharmacogenetics. I want to bring this up, because I want to bring this back to aging. We've been talking about it, but here's a more direct focus laser beam to it. 7 years old, this paper, but okay. It's not a problem. Because we're going to talk about a paper a lot more recent. Now, what are some clinical applications of this whole discussion? Well, you know about pharmacogenetics, right? Tailoring or targeting drugs for specific genome of your patient, okay? SIPs account, wait a minute here, SIPs account for 80% of the variation of metabolism of pharmaceutical drugs. Now, that's not a surprise when you just looked at all those tables I showed you. 80% of all the variation of the pharmacokinetics of your drugs come from SIPs, okay? Wow. All right. Now, variations in SIP sequence account for the difference in SIP expression and biological activity. The whole ball of wax here is involved, right? How much SIP is made, how potent is the enzyme activity, is it in the right cell at the right time, all of that. SIP genetic variation, and there is, of course, genetic variation in these genes, right? Going to happen. Correlates, it can correlate when you get genetic variations such as single nucleotide polymorphisms with therapeutic failures, they call these TFs, and adverse drug reactions or ADRs. Okay, so when you have overpotency of a drug or you can't get good dosing of a drug, if you go in and you look at a patient's individual CYP levels, you might find a reason why, because they're not expressing the right CYP for detox, or they're overexpressing a CYP which could enhance intoxication or prolonged activity or circulation time for a pharmaceutical drug. Okay, so you get the idea there. So ADRs, okay, what are ADRs again? Remember, those are adverse drug reactions, are responsible for morbidity and mortality, and you see this increasing with older or more elderly patients. Drugs are more potent, drugs can have more side effects or negative effects, and that's because of this adverse drug reaction. And we just told you that that variation in the ADR and in therapeutic failure can follow the variation in mutation and an alteration of expression of cytochrome P450 oxygenases. So that means that a lot of pharmacogenetics in the elderly is starting to look at isoform families of CYP P450s in that population to tailor specific drugs to individual patients. Okay? Which, of course, there's some obvious is some inheritance here for the mutations and for the sequence of specific nucleotides and specific genes for all those cytochrome P450s from inheritance. But we also know that there's an epigenetic component. And lo and behold, here's a paper. Genetics aside, what are the epigenetic components of therapeutic failure and adverse drug reactions? Here's a paper published. This was now only actually a little over a year ago, right? Because April of 2017, I'm speaking right now. So 1 year and 4 months ago, maybe. Epigenetics of aging and Alzheimer's disease, implications of pharmacogenomics and drug response. So there you go. One of the major diseases of aging, right? 50% of all people that get over 65 years old, ultimately, if they keep on living every 5 years, they're going to enhance their probability of getting AD, Alzheimer's disease, right? The canonical late onset Alzheimer's disease. Well, what if we look at the epigenetics of that aging process in that disease cohort? What people are finding in terms of trying to target pharmacogenomics is that a lot of the genes that are altered and a lot of the expression of those genes that look unique to the aging AD population are cytochrome P450 monooxygenases. Okay. Thank you very much. I wanted to make that as quick as possible. I know you probably have a lot of other things to do, such as like go back and read this and watch this a couple of times and really enjoy it and learn hopefully something. But also, it's just good to end things so that the next time we do it, they're just that much more fun, right? So you can contact us at that email there. Please do. We would love to have questions from you, and we'd love to work with you in your biomedical questions. And we will go into the literature. Like here, I'm doing it on my own. I'm just investigating to show you the kinds of things that's in the literature that you may not know. But we will tailor all of these investigations directly to your questions. There's our website. And again, we're scientists verifying published evidence in medical biosciences. Okay. Thanks a lot for your attention, and please have an authentic day. Bye.